De volgende spreker is Isabel Siles. En Isabel Siles is een PhD-kandidaat in Environmental Biology. En dat doet ze bij het Centrum voor Milieuwetenschappen van de Leiden Universiteit. En zij onderzoekt hoe de bodem de kwaliteit van de gewassen en de gezondheid van mensen kan verbeteren. Ze doet dit samen met een interdisciplinair team in het Soils2guts project. Ik denk dat we daarmee gaan starten. I just introduced you. Welcome. Go ahead.
Thanks for the introduction. Well, as India mentioned, I'm going to be talking about the Soils2guts project, and our aim is trying to understand how soil microbiomes are connected to human gut health. So, as Dan mentioned, with population continuously growing, we have relied on agricultural intensification in order to ensure food security for us all. These includes practices such as increased use of fertilizers and pesticides, heavy machinery, but also the use of high-yielding crop varieties and long monocultures. And we are now very aware that this could lead to environmental deterioration, such as biodiversity loss, also pollution of water bodies, and as well as soil erosion. However, it is often overlooked that these practices have also led to a focus of quantity over quality. So, as Dan mentioned, like, we are not looking into how nutritious the crops that we're growing are, but more of how much yield we can provide, and that's what the system is promoting. Here is where we think that soil microbes could play a very important role on improving crop quality. And as you can see in the picture, when you have a very healthy soil, what the plant does to grow is recruit microbes into the roots. Then these microbes can help the plant protect against pathogens, but also they can help mobilize nutrients and solubilize them so they can so the plant can use them to grow. We think that these in turn could directly help and improve the plant microbiome, and both the plant microbiome and the soil microbiome can indirectly improve plant nutritional composition. And the thing that we want to see is how can this be linked into a balanced gut microbiome and an adequate gut health. So what happens when your soil doesn't have these beneficial microbes to be recruited? Well, we think that then pathogen invasion is much more common in these crops, as well as there's not enough nutrients to be solubilized and mobilized into the plant, which would lead to avoiding all these processes and could lead to a perturbated gut microbiome and impaired gut health. So in order to tackle this puzzle, we decided to come up with the Soils2guts project, and each team has its own expertise. I'm part of the soils teams, and here we're gonna see how agricultural practices affect soil microbiomes. Then I closely work with the plant team, where Marike, my colleague, if you have any questions, you can also go to her, works with plants and how the soil microbiome can affect the plant growth and nutrient quality also. Then we have the human side. Here we have both people working with human interventions, but also with ex vivo models. And finally, overviewing us all, it looks funny, but it's Shana, and they are working with governance and seeing how these systems can be changed and how all this knowledge that we're getting from the project can actually be applied into policy. So the first step in our project was trying to see how agricultural practices can affect the soil microbiome, and we need to establish this baseline of how crop microbiomes relate to plant quality. In order to do so, we sampled 31 farms across the Netherlands. We had 19 carrot fields, 15 potatoes, and 14 lettuce. In each farm, we asked the farmer a questionnaire from which we will get to know all the practices, what fertilizers they use, about pesticides, cover crops, crop rotation, etc., to calculate a land use intensity index. We also took both soil and plant samples. And from these samples, we wanted to analyze the microbiome and functional diversity via DNA extraction. And finally, we want to analyze plant quality via nutrient content and also metabolites, which I will go further in the presentation later. So what do we expect, or what do we expect to see? We think that, and Dan mentioned it, it's not precisely one set or one category of agriculture that is going to have the highest soil diversity, but in fact it's going to be in a gradient. So the more intense your practices are, the lower your soil diversity is going to be. So it's going to be the higher your intensity in land use, the lower your soil diversity. Whereas in contrast, we think that the higher the soil diversity of the higher the plant microbiome, or the more diverse plant microbiome you're going to find, and this also is going to relate to plant quality. So both a higher plant microbiome will be related to a higher plant quality, but also a higher soil diversity would have an indirect effect to plant quality. So then I'm going to be showing some graphs, and you can follow it with our findings so far. So the first thing that we had is how does microbial diversity correlate with land use? Here, well, it was supposed to be orange, but I think it's red. So the colors are going to be the same in all the graphs. So red, orange is carrot, then lettuce is green, and potatoes are blue. And in your left side, you have bacteria, and in your right side, you have fungi. Here on the x-axis, we have the farms that we sampled, so each column is a different farm. And on your y-axis, you have richness or abundance, the number of different bacteria and fungi that we found in our samples. And as you can see, there is a very, regardless of crop, there is a very consistent gradient on how diversity is correlated with the farm. So it's not necessarily correlated to the crop, but it should be like the agricultural practices are driving this type of gradient. Secondly, we want to see how soil diversity is connected to plant microbiome. In this graph, we're just looking at bacteria, and if you remember for the first figure I showed, we know that plants recruit microbes into the roots, right? So the soil that is very closely attached to the root is called the rhizosphere. So this is what we're measuring. We just don't want to see what is in the soil, but actually what the plant is recruiting. So here we have rhizosphere bacterial richness, so the amount of bacteria that we found in the rhizosphere. And then we also measure the edible part, bacterial richness. So from each plant, potatoes the tubers, and then, for example, lettuce the leaves, how much bacteria we found on them. And as you can see, each crop had its own different correlation with it seems that lettuce was the one that had a bit of a higher correlation as we expected it to have. Carrots also followed kind of a similar pattern, but actually potatoes were not what we expected to see. And then on the third part, it's what we are currently working on, and it's how to measure plant quality. I think Dan mentioned it very, very nicely. It is very hard to determine or to give a grade to how good our crops are. For example, you can have physical measures of quality. It could be texture, color, or taste, but these kinds of be subjective, right? So there's also chemical measures of quality, and then you have micronutrients and also micronutrients. Dan is very focused on this, and we want to work on measuring micronutrients too. But sometimes it is a bit harder. As you saw, he measures so many nutrients. So we also think that we can go a step beyond that, and we can measure secondary metabolites. So what are secondary metabolites? Basically, they are organic compounds that the plant produces that are not directly involved on plant growth, but they give the plant some benefits while they are growing. For example, carotenoids. We know that in plants, they can help with photoprotection, pigmentation, and photosynthesis. But why would it be interesting to measure them? Because we see that they have a benefit on the plant, but they can also have health benefits. We know that they can decrease the risk of several diseases. they can prevent retinopathy, and they can even help in both health. So that's why we think these type of compounds would be interesting to get measured. And carotenoids is just one of the examples, but we're looking at several one of them. So right now I cannot show you the perfect graph that we want to see, but we do expect to see that if the plant has a function for this type of compounds, we definitely are going to see some type of correlation with soil diversity. So what can we do with all this information? The fact is that both the agricultural sector and the healthcare sector have very systematic problems and challenges, such as environmental deterioration, but also chronic diseases. So we think that the system needs to change. The current system that we are living on needs to change. And in order to do so, we need to change policy and also a common public mindset. On the work that my colleague Shanna has been doing, she has noticed that there's a lot of common ground that we can exploit in order to promote this change. For example, farmers are open to produce healthier and more sustainable food, but the system needs to compensate them. So yeah, we might need to shift a bit from just getting the food more expensive, but actually promoting that they get these sustainable practices into their land. And also healthcare professionals recognize the role of food quality in long-term health. However, there's still a lot of ongoing barriers on promoting this change, and it could be because there's a lot of limited and fragmented collaboration. So it is really hard, in this project we can't even see it, to match all even the language and the goals and overall in the government system, the incentives in order to get this change going. So I know I talk a bit about, and it's a very broad overview of what we're doing now, but I just wanted to give you some take-home messages. The first one from the first graph is that we see that agricultural practices lead to a gradient in soil microbial diversity. So again, there's not a good or bad or not just a right or wrong answer, but we see that all the combination of your practices can lead to a more diverse soil. Secondly, that soil microbes actually correlate differently with plant microbiomes. So besides the diversity that you saw in the soil, the plant is going to react differently on and recruit differently the microbes into the roots. Finally, we also think that secondary metabolites are a very good option as a plant quality metric because they have both functions in plants, but they can also provide a lot of health benefits. And finally, we need to combine efforts in both the agricultural and the healthcare sector to change the current system that we're living on. So what is in the future? I know I haven't presented as much of the healthcare part because they are still going on that. So first, they want to test some of the specific organic compounds, as we mentioned before, the secondary metabolites on synthetic gut communities and models. This year, we're also going to select fields with very distinctive microbes, and then we're going to grow food on them and do a human intervention to investigate health outcomes. And finally, the overall arching goal of the project, which is finding key taxa or these key role players in the soil, then that you can see how to steer the existing soil microbial community to increase soil functions, and these could later lead to improve plant quality. So yeah, if you want to keep on looking what we're doing and keep up with all our updates, you definitely should check our webpage and also our LinkedIn. And if you really want to talk about soils, this is my contact information. I'll be more than happy to get in touch with you. Thank you.
Thank you so much. Very interesting. Thank you. I'm very surprised that the soil has such a different influence on different crops. I think for me that's a new thing. Thank you. There's time for questions. I see first hands coming up. I'll start here in the front. Please, your name
Hello.
and organization before you start asking
Yep.
the question.
Hi, my name is Melchior. I'm from Nimble. We're setting up a processing facility to connect local sustainable farmers to the B2B market. And my question is, you started off with also measuring land intensity, usage of farmers, and then afterwards you scaled them. You put all the farmers next to each other in the columns. Was there any correlation between land intensity and the results in that farm? Because there was nothing on the x-axis. So I was curious if there was that correlation.
Yeah, we're working on the score. So right now I just wanted to show you there is a gradient on diversity, but basically what the land use intensity index is going to be, it's, a score. It is really hard to give scores to farmers, and we saw that with nutrient density. What we were trying to do is give each farmer a score of how intense the land use that they, like the practices that they are having, it's on the land. So that's what we got from the questionnaire. Basically, size comes into consideration of their farm, but also the amount of pesticides they're applying, organic fertilization, mineral fertilization, if they live cover crops or not, the rotation that they have, and even we want to include if they have legumes on the rotation that could actually improve soil microbiome. So we are basically working, it's a lot of data and it's a lot of categorical data that we basically want to change it into give them a grade. And we expect then that would be the axis in the future. Like, we're still working on it, but that would be the axis on the x-axis. That would be the grade. So basically the land use intensity index, and then we expect to see some sort of correlation with soil diversity.
Thank you. Other questions? And you can turn your perfect. I saw another hand. Yeah, up there.
Ik ben Irene Voskamp, province of South of Holland. We have a big greenhouse industry in the south of Holland, and it is, has many advantages. For example, in relation to water necessity for the growing. How does that relate to this discussion? Because they are obviously not all, the greenhouses obviously don't always use the real soil under their greenhouses.
Yeah, we also actually sample some greenhouses during our field season, and it depends. We are thinking of them about as substrate. They are also going to get a score, but usually greenhouses, I mean, it has their advantages and their disadvantages, and within greenhouses there's different practices, so they are going to get a score, but we think that they are a bit more intensive in the sense that they are not actually just growing in the soil, but they are definitely going to be included, and I think there's a lot of room of improvement on how to minimize the intensity of greenhouses.
Any other? Yeah, I see it here.
My name is Marius Moner. I'm from Avans University of Applied Sciences, and in your second take-home message you said soil microbes correlate differently with plant microbiome across crops. And then I wondered, do you see different correlations between monocrop in rotation and polycultures, how they affect the microbiome?
Actually, each farm had it different. We have both monocultures and also intercropping, and we didn't see any trend on that. So yeah, we really didn't see a correlation between, like, I mean, we're gonna get the practices overall as a score, as a land use intensity score, but I think it would be very interesting to see if there's actually some correlation between the ones that had intercropping. I mean, we have it as yes or no, but I don't think we have checked on it, but I'll definitely will take a look into that.
Nice.
Here, vooraan.
Hi, I'm Kirsten Berg from the Erasmus Medical Center in Rotterdam. And I'm a nutritional scientist, so I was very interested about the effects on health, of course. And I know you love to talk about soil, but perhaps you also love to talk about health. You were showing future plans where you want to grow crops on different types of soil and then also have a human intervention study. what kind of biomarkers for health were you planning to look at as outcomes? Because I believe that the changes would be rather small, and perhaps you will need like a very large sample or a large amount of time. How do you plan that?
I'm gonna tell you what I know of the project, so I definitely can get you in contact with the people in charge of it, but I think there's gonna be two different trials. One is gonna be with I think with university students, and then it's gonna be like they are gonna give them the food, and then a week after and see how it changes their gut microbiome. I don't think it's health markers. Maybe that's my bad
So
on
it's
it. It's
gut
more like how they
microbiome.
change the gut microbiome. And then, but for health markers, they are gonna do a similar study, but with patients that are in the hospital for how do you call irritable bowel syndrome, I think, or
Okay.
some type
Yeah.
of a
Yeah.
gut disease, I'm gonna say, and then I think that's where they are actually gonna measure if it actually improves their
inflammation
health.
markers,
yeah,
perhaps.
yes.
Yeah.
But
Yeah.
I cannot tell you specifics, but I know those are the two human intervention trials they're gonna have, and also they are gonna do it with the gut model.
Okay, thank you.
Ja, ik zie hier nog een vraag. Mag ik de microfoon even aandoen?
Ja, mijn naam is Eline van Hoogdalem. Ik ben I'm a volunteer, a food forest volunteer, and hopefully and also a candidate for a food forest. And can you tell me more about the gut, this artificial gut you use for testing, or did I understood it not correct?
Sorry, what do you? I.
She asked if you said something about artificial guts.
Oh yeah, the
Yes,
gut
yes,
models.
yeah,
Yes. Yeah. Again, I'm not on the health part, but what I've understood is basically they have an artificial model, basically like an intestine on it, and they are isolating a specific of the secondary metabolites. I know they are working right now with glucosinolates. Our postdoc in the health part called Diana, I can give you her contact information, and they are seeing how introducing these isolated compounds can affect a synthetic gut community. So it's imagine like a synthetic gut model, and then that's where they put the glucosinolates and they see how the gut community can change.
Synthetic via the computer model. Is that the case?
No, I think that it's an ex vivo model, so it's a physical thing. But I'm not super sure how it works. I'm not sure how it looks. I bet it very cool, but I am not sure.
Ja.
Okay, thank you.
Well, also, I saw some people... I'll do it in Dutch. Ik zag wat mensen die foto's in het maken waren ook van de slides en de contactinformatie. En weet dat we de slides ook delen. Dus jullie krijgen alle contactinformatie en dan kunnen jullie eventueel nog vragen stellen. Ja? En zou je microfoon... ja, dankjewel. We have time for one more question. Let me see. Oh, I see one there. Yeah.
My name is Robina, and I'm orthomolecular therapist, and I have also a question about the health part. Does the team of the health part has expectations about what they will see on the crops? What kind of microbiomes? And does the results fit their expectations?
You think what they see on the crops?
Yes.
Actually, I guess that would be analyzed by the plant part. But what we're doing for the experiment with the human intervention, it's not that much of what we found on the plant, but what we found on the soil. And I mean, we are seeing that there is a correlation with the plant microbiome, but I don't think they are looking into a specific group or genera of microbes on them. I don't know if that was your question, like if we're looking into a specific group of microbes that we want to find in the crop.
Yes, but you thought about on some crops there was differences between the other crops, and does this was you surprised about these
Yeah, I think we were, and maybe what it's something that we're gonna be looking at. It's looking for crops that are eaten raw, because then we expect them that the microbiome in the plant is not gonna change. So definitely they are looking into those types of crops, but I don't think we're specifically looking into one set or one, let's say, ideal plant microbiome in order to feed.
Okay.